This experiment was conducted to answer the question, how is the height of mung beans affected by the elements of the closed system? The results would demonstrate how the factors that allow the growth of plants can be recreated inside a terrarium. Additionally, if this can be done in a closed system, then humans might stand a chance of survival against themselves.
To build this terrarium, we began research. From gardeningknowhow.com, we learned that mung beans require sunlight and water, along with soil that's absent of rocks. We also learned that ecosystem processes such as decomposition are vital to biogeochemical cycles, as they are the part that returns phosphorus, carbon, and nitrogen back to the soil to be reused. Additionally, it requires a dark, damp place so that biotic factors like bacteria and mold can function better, according to moldbacteriaconsulting.com. These cycles would be different, however, because our terrarium would be closed and certain steps in the cycles would not be included, like marine life or precipitation.
From Instructables.com, we learned that to make a terrarium, we need to cut the bottles as shown here, and add 1 cup of rocks in each chamber so it fills about 7.6 centimeters high. Add 3 2/3 cups of soil above the rocks in the terrestrial and 1 cup of soil in the decomposition chambers. In the decomposition chamber, we added 2 cups dead matter, and the aquatic chamber 2 cups of water, and 3 mung beans placed 1 inch deep into the soil of the terrestrial chamber. Each cap will be punctured 6 times in order to allow the flow of nutrients, and the whole terrarium would come together like so and be taped. A string would go through each of the chambers to utilize capillary action to let water in the aquatic chamber move upwards. The decomposition chamber would be in the middle since it is dark, the terrestrial on top because it is bright, and the aquatic on the bottom so nutrients could collect there.
The original prediction of the experiment was that if growth in the mung beans are observed in addition to algae in the water and a consistent water line height, then the terrarium is successful at maintaining biogeochemical cycles such as phosphate, water, carbon dioxide, and nitrogen, along with having abiotic factors such as sunlight and temperature. Therefore, the closed ecosystem has terrestrial, decomposition, and aquatic chamber that supports each other.
The hypothesis is supported by the data because plant height increased overall with 3 notable bursts of growth. Within the first week, the average plant height remained at 0 centimeters until day 4 when it rose to 1 centimeter. After day 9, the stagnant growth explodes until day 13 when the plant height is 9 centimeters. The height stays this way until day 16 and the plant grows to an average of 14 centimeters by day 20. Additionally, water height overall decreased minimally by less than half a centimeter, with 2 losses of water between day 4 to 7 and 9 to 10. The height began at 12.7 centimeters until day 4, where it dropped to 12.4 centimeters by day 7. The water again decreased by 0.1 centimeters over day 9, and by day 10, the water reached a constant height of 12.3, where it remained for the next 10 days.
Possible reasons for the growth in plants would be attributed to sunlight, carbon cycles, and water cycles. According to Khan Academy, plants need sunlight, carbon, and water in order to properly photosynthesize or gain energy. Mung beans are plants, thus they too require these materials to grow properly. Due to the fact that the average height went from 0 centimeters to 14 centimeters in 20 days, the sunlight reaching the plants was sufficient for the plant's growth. The second requirement for photosynthesis is carbon, which the plants could be getting from ecosystem processes like decomposition in the decomp chamber, as the mold observed in day 4 breathes through cellular respiration. Additionally, the mung bean shells fell and began to mold, demonstrating a miniature carbon cycle in the terrarium by releasing carbon dioxide. Thus, the carbon cycle is evident within the terrarium. Lastly, the plants receive water from the string connecting the terrestrial and aquatic chambers. This is because as the water level dropped, condensation was observed in the upper chamber on day 6. Since plant growth was observed, sunlight, water cycles, and carbon cycles must have been adequately supplying for the plants.
Water height experienced a total decrease in water level in graph day 0 to 12 of about 0.4 centimeters. This decrease could be attributed to equilibrium, plant absorption, and capillary action. Equilibrium is how water evaporates until there's too much water in the air, so some condensates and more evaporates. However, the total amount in the air and in liquid form remains constant according to hyperphysics.edu. This can be observed in the graph days 10 to 20 where the water stays at 12.3 centimeters. Thus, the water must have reached an equilibrium in order to stay at the same height. Due to a phenomenon known as capillary action, the string drew water against gravity up to the terrestrial chamber throughout the duration of the experiment. This water was absorbed by the plant's roots in order to photosynthesize and grow. Some of it was then transpired into the air and condensed on the walls, which could fall back down only to be transpired again since water droplets were visible on the top of the terrarium on days 3 and 6. Due to the fact that the water cycle reached an equilibrium and cycles around the terrarium demonstrates a sufficient water cycle capable of maintaining plant life.
The nitrogen and phosphorus cycles were also present, as seen in the decomposition chamber. The mold observed on day 4 shows the return of nutrients like nitrogen and phosphorus to the soil. Miller, Levine, 538, which slowly leaks into the aquatic chamber. The Michigan Environmental Education curriculum says that algae needs some phosphorus and nitrogen to grow. Due to the fact that algae was observed in days 13, 18, and 19, the nutrients from the decomposition chamber must have been the result of phosphorus and nitrogen cycling around the terrarium. Consequently, terrarium has working phosphorus and nitrogen cycles. Since the terrarium has successful biogeochemical cycles and abiotic factors, the different parts of the terrarium support each other.
In the data, there are multiple causes for inaccuracies. The first inherent cause regards the daily water level measurements. As nutrients from the decomposition chamber fall through holes in the cap of the chamber, small particles of dirt and rock also fall through. This affects the data by making water measurements higher than it would be without the added particles. A future extension to mitigate this issue is to add a coffee filter in the cap so that water and nutrients can flow through but soil cannot.
One human error is centered around the plant height. The seeds were planted too low and some of them were spaced too close to one another. This error affects the data by making the average plant height lower than it should be, and hinders effective data since the two bean sprouts close to each other may compete for resources, green ecology number 2, and have lower heights than the lone sprout. A further extension related to this is to stick the ruler into the soil to gauge the depth as we drop in the beans.
Furthermore, our bell schedule is a contributor to human error. The normal time of measurement from one day to the next is 24 hours. However, on Tuesdays to Wednesdays, the time is 24 hours and 30 minutes, and on Wednesdays to Thursdays, this time is 23 hours and 30 minutes. This makes measurements on Wednesday slightly higher for plant height than they should be, and measurements on Thursday slightly lower than they should be. A possible solution is to always take the measurements on a set time between 11:42 and 12:11, as we are always in the classroom at this time regardless of day. This acts as a variable extension by removing the additional variable of the bell schedule.
The final extension that will be made is the use of a heat lamp rather than the sun. This is because the sunlight that the plants receive is different every day as it depends on the weather. Although these differences better model natural growth, for the purpose of this science experiment, it would be more beneficial if a consistent light source like a lamp was used. As a result, the growth of the mung beans would go up on cloudy days to match days where it would be sunny, so that the resources available to the plants are consistent.
The purpose of our terrarium was to answer the question, how is the height of mung beans affected by the elements of the closed ecosystem? The key finding was that the mung beans grew, thus the biogeochemical cycles, ecosystem processes, biotic factors, and abiotic factors in our terrarium could support the growth of the mung beans. In terms of plant growth, we believe our terrarium was successful as the average height of the plants, our dependent variable, was increasing as time, our independent variable, increased. The terrarium lasted as long as it did because as the plants used up ingredients during photosynthesis, these ingredients were eventually returned to be used again. Our main modification, the string, allowed additional water that was not initially added to the terrestrial chamber to water the plants, which enhanced the growth of the mung beans. We should care about building terrariums because they can function forever without drawing resources from anything but the materials inside. If humans could model this, then we could live on Earth for generations to come since Earth's resources would be reused with absolutely no need to get more resources from other celestial bodies.